is we can go back to our original R2 Genomics. And if I want to find something, I want to find out what A2ML is doing, what we can do is go back to the main. So left-hand corner of your window. Go single data set, tumor breast cancer. We've got this should still be up on your thing, right? Now instead of saying view a gene, what I want to do is I want to say find correlated genes with a single gene. So it's about, I don't know, a fourth of the way down. Does everybody see that? Click on that. And then hit next. Just like I told you, again, remember we can do class comparisons. That's what we did at first. We found out what was different between ER positive versus ER negative breast cancer cells. I found the gene that was at the top that most differentiated them is this, alpha gluba, whatever what's the name of the gene again? Alpha 2. Yeah, okay. Basically, I have that gene, it showed up. I looked to see if it had any relationship to breast cancer, couldn't find a lot. I think this might be a really interesting target to look at. Maybe I'll get a grant on this, right? No one's really looking at this area. So here's what we can do is up here on Gene Reporter, put that gene, A2ML1. And what you'll see is it'll basically, a gene will come up, you can just select that. Okay, once I've put it in, I can go submit. So what I'm doing now is it's going to find this gene's best friends and worst enemies. If I know what its friend's doing, I can kind of figure out what it's doing. If I know what its enemy's doing, I know what it's probably suppressing. Does that make sense? So here we go, and basically you've got positively correlated and negatively correlated. Here's what I can do, is on the right over here, see these brown things? What I want to do is save selection as a text file. Save current selection as a text file. Select. Actually, let's get rid of the header. So here's what I'm going to do. Go back and say, instead of saying save current selection as text file, save selection as text file, no header. Do you everybody come up with a strip file link? Click on that. Okay. Here's what I want to do is, now that I have this, I have my list, I want to save this as a text file. So in your browser, under File, go Save As. Save Page As. And I'm just going to put it on my desktop. I'm going to label it A2ML1. Hit Save. Now, wherever you saved it, show it in your Finder. And what you're going to want to do is open it with Excel or some other kind of... Where the hell did it go? Oh, there it is. So here I'm going to right-click. I'm say open with Microsoft Excel. Everybody do that? Does it look like this? Everybody's good, right? We have a list. It has our probe set ID. It has our gene symbol on it, and then it has an R value. So this is your value of, and then we have a p-value associated with that correlation. Here's what I want to do is I want to find all the things that are positively correlated to it. So what I'm going to do is I'm going to put my cursor into C2, and then I'm going to sort largest to smallest. Yours might say sort descending. You should have A2 ML1, and it should be your top hit. So here's what we're going to do. So just watch. Like I said, it's recorded. I've got lots of videos over here. So here's what we're going to do is I'm going to take the top 250 genes. So again, I've sorted by R value. I'm going to put my cursor at B2, and I'm going to go down to 251. Because we've got a header. So I want 250 genes. So on my list, it's FOLH1. Select all those, shift select, and then copy. Anyway, command copy, control copy, do it off the menu, whatever you want. Everybody do that? Okay. Here's what you can do is you can go to this website though. And what I want you to do is, again, remember, bookmark all these different websites. Let's go back to our original browser. And here what I want to do is I want to put string, and then you can go protein as well, so you can find it. I hit return. So you should see string-db.org. That's the one you want. One time I was showing this to a high school class, and we were trying to use string, and so I typed in just string in the web browser, and all these pictures of string bikinis came up. I'll tell you what, that high school class was definitely like, they were zeroed in after that. Science is fun. Yeah, I know. Okay, so here's hit stringdb.org. So it should look like this. Actually, you'll get to there'll be like a landing page. Say you want to search, and then you should get to something like this. Everybody there? Okay. So here's what I want you to do is I want you to make sure you're on multiple proteins. This is how easy this software is. So here's what I'm going to do. I'm just going to, on these multiple proteins by names, I'm just going to take my list and I'm going to paste it in. Everybody do that. That is able. Okay. Then what you're going to do is you're going to want down here, you're going to, for organisms, select Homo sapiens. You have to select it, and then what happens is like it's at the bottom, you have to like go select. All right. Then we're going to hit search. This is just telling you what genes that it recognizes, right? You can go in here and look at any of these different things. We've got 250 query items. Now I'm going to hit continue. Here's where the magic happens. I love this. This right here is A2ML1's gang. Again, this isn't a canonical pathway. This isn't something somebody defined. What I did was I went in there, we had lots of samples. We had over 3,000 samples to choose from. And I said, find me genes that look just like this other gene. Because if they behave the same way over all those samples, they're probably involved in similar processes. I would much rather look at a gene list like this than in a table. Does anybody understand why? What does this give you? If I'm looking at this and I'm going, what are my key players? What do you think I'm looking for? What would you look for? Again, say this is like a organizational, like, you know, like something they'd put on the cop shows, you know, this is like a diagram of all the criminals, right? Who am I looking for? Who's going to be the most important criminals in here? Yeah, or basically the ones with the most connections, right? And I can totally download, if you go down to like here, exports here, I could export all this information and I could basically search which of these genes has the most connections to other genes on my list. We could also try to find ML, basically find the gene and like pull it out. Does anybody see do you see the A2ML gene at all? Where's it at? And again, you can take these, this is Java, so you can take it and you can actually move these things around. Move things. Here's what's really interesting. If I go down here, so if I go down, I go to settings, go all the way to the bottom, and what you can do is you can find, let's see, hide disconnected nodes in the network. And then I can update it. So now we're only just looking at things that are connected to other things on our network, which didn't eliminate a whole bunch. Like this gene right here. This might be a godfather, right? Look how many connections it has. And it's a transcription factor. So again, it's going to influence a lot of other genes. Again, what this allows me to do is if I find the key players in here, I can basically find out like what's actually doing in this cell, find its best buddies, and then maybe he's got like I can find a snitch in here that's going to allow me to manipulate, bring down the whole system. Does that make sense? We can also color it. So we can go down here. I can go to analysis. So we put 250 genes in. It recognized 248 of them. That's pretty standard. It expected, based on what we put in, it would expect that we would get 231 connections by random chance. We got 857. The odds of that happening are 1 times 10 to the negative 16th. What I'm trying to tell you is what we pulled out of here is a network for sure. The odds of me seeing all these connections by random chance are very, very infinitesimally small, which would tell me that whatever we're pulling out here is absolutely connected. Like we pulled out a signaling network. And again, I didn't need a bunch of experts 30 years ago to tell me what a pathway is. We found it on our own. And if we start looking at some of these genes, we can see that anybody here of aurora kinase? Very famous oncogene. It's in there. What we can do is we can actually start coloring it. So again, this is his network. So I'm going down, I'm under the analysis tab. I look here and I can see all these different kinds. What do you think? Where are these genes involved in? What do you think? Cell cycle, for sure. Cell cycle checkpoints, sister chromosome segregation. We can go down here. Where are these, where are they located at? A bunch of these, more than we would expect by random chance, are associated with chromosomes, centromeric areas, kinetic cores. This is absolutely, whatever A2ML1 is doing, it's absolutely associated, at least in breast cancer, is associated with, with cell cycle. And then here what you can do is you can get to different, basically, papers which also pulled out some of these genes. And when you start looking at this, cognitive emotion, salivary alpha amylase, cyclins. We can go down, we can look at some of the clusters that the actual program has itself. Again, a lot of spindle. Not only is it cell cycle, but it seems like most of these are involved in separating the proteins and also involved in cell cycle checkpoints, which is absolutely critical for cancer. And I can absolutely color those as well. We're also seeing retinoblastoma gene in cancer. We're seeing that gang kind of shows up as well. Retinoblastoma is a very well-known tumor suppressor. It's a COP. And again, this is showing up here as well. Again, we can come back here. We can basically download any of this information. I can download the picture. I can download the gene list. I can download where the connections are. I can basically blast that and figure out what genes have the most connections to other genes. And that's what gives me an idea of what A2ML is doing. And at least in breast cancer, even though it's a globulin gene, it absolutely and utterly is associated with cell cycle for sure. What I could also do is I could color it and I could say which ones are, based on what I labeled it, which one has the most colors. And again, that Aurora kinase B shows up as well too. The rock star of cancer. This is big time criminal. Come on. And it seems like this big knot here seems to be associated with cell cycle. But I bet if we went up here, like SOD2, does anybody know what SOD is? Superoxide dismutase. It's an antioxidant. We probably got an antioxidant kind of little pathway in here as well, right? It's not just cell cycle, but we can find these little notches. Then if I thought, oh, SOD2 seems important, I could then go correlate gene expression on SOD. Does that make sense? What we could also do too, if we wanted to get more information, and I want to show you this website as well. So be sure to bookmark String. String will be your best friend. Because number one, there is not a lot of networking, basically platforms out there that allow you to do this, put genes together like this. Not only does it do the networking, but it also gives you an idea of the overrepresented biological processes associated with that. So you get like 2 for 1 off this deal. You get your networks, you see what's interacting with what, and then you can also basically see what those genes, based on all those genes that are associated with your gene, you could see what they are doing. If I did the same thing to the negatively correlated genes, we would see something completely different. Does everybody understand that? Right? And what this would probably be is this is inhibiting whatever is negatively correlated. It's that yin and yang, right? When this goes up, this other thing is going to go down, and vice versa. And this also seems to be associated with the ER status. So estrogen signaling is absolutely important in there. When I did do the negative correlation, what I ended up with was a bunch of growth factors, right? These are both cancers, right? But in this particular cancer, instead of using growth factors to drive transcription, it's using these genes to do that. Does that make sense? Very, very good program. I love String. I've reached out to them as well. Again, when you go to the site, it goes, welcome to String. It's just so nice. All right. Oh, here it is, A2ML. Here is what is very, here's what I want to show you is like, we just generated a complete pathway and network for A2ML. But based on what is known in the literature, these are the only connections that it comes up with in its database. I took its gene, right now its gang is like 3, right? And these seems to be keratin related things, which is associated with cancer. usually associated with a particular cell type. But again, I took it, I expanded it out, and now we're out to this. And again, if I want to look at any of the interactions here, A2ML, what is the basis for them connecting these two? I can click on that connection, and this will tell me what evidence it's using. All right. Okay, we've got gene fusions, no, co-expression, no. Experimental biochemical data, putative homologs were found interacting in other organisms. Right? And they're also, these two genes were mentioned in a PubMed abstract. That's how it's connecting those things. The thicker, the more lines you have to different genes, the more evidence for a connection. Does that make sense? Again, I get so much more information looking at genes this way than in a stupid table, black and white table. This is how you should be looking at this stuff. Here's what usually happens. People, scientists go out there, they'll do this t-test, they'll come up with a gene, and then basically they'll do the t-test, and then they'll just look at the gene that's expressed that has the highest fold change. And it's not necessarily going to tell you anything, right? Maybe you on that end, you just got a thug, right? And this thug just seems to be really active, but it's probably being bossed around by something else. And that's what you really want to get to. Okay. So let's look at one other thing. Here's what I want you to do. I want you to put in human protein atlas.